All right, good afternoon, everyone. My name's Austin, and I'm a master's student at the University of Oklahoma. I'm really excited to talk to you all about my research, but before I get into it, I want to give a little bit of background and setup so you know why I am doing what I am doing. So animals live in a microbe-rich world, and microbes are ubiquitous, and they're routinely interacting with animals in ways that are both positive and negative. And these interactions span a wide variety of taxa across all domains of life, and these are just a few examples. So for example, leafcutter ants, many species of them, sort of cultivate these fungal colonies within their fungal gardens within their colonies and provide them fresh leaves and then harvest the fungus. Hawaiian bobtail squids interact with the marine bacterium Vibrio fischeri and produce the squid's bioluminescence. Also, rhizobia and legumes collaborate in these complex series of interactions to form these nodules that allow them to fix nitrogen. And there are many, many, many more examples of this, although I don't have time to talk about them, but it's important to note that these interactions are important for life as we know it. So the evolution of these interactions, however, are areas where our knowledge has gaps. And our lab is asking questions about these gaps. So how do microbe-dependent life history responses evolve in divergent hosts? What are the genetic and molecular bases of host-microbe interactions? And how do microbial communities evolve as their hosts evolve? And to be able to address these questions in an efficient manner, you need a system that's conducive to doing so. And we've opted to use Cinerobditis nematodes as our system to do this. Cinerobditis nematodes, especially C. elegans, have been used for 50 years now because of a variety of traits that make them very appealing for research. They're really cheap and easy to rear and maintain on agar plates. They're bacterivores, so all you have to do to feed them is pipette some cultured bacteria onto the agar plate that you put the nematodes on. They're genetically amenable, meaning you can do things like CRISPR with them relatively easily. And additionally, from the time of hatching from an embryo, depending on what species you are, these nematodes become reproductively mature in like 2 to 6 days, so they're extraordinarily quick, and you can also get a lot of nematodes fast and use a lot of nematodes for your experiments, meaning it's high throughput system. However, until recently, not much was known about their ecology, and traditionally these worms have been raised on E. coli in the lab. But this is not what these worms eat in nature, so consequently, studying what these nematodes actually eat in nature and using their power as a model system is it makes it an ideal candidate for studying host microbe interactions. And additionally, we've opted to use a recently discovered species of Cinerobditis, C. inopinata, and C. inopinata is the sister species to C. elegans, and you may wonder, why is this important? Why do we care about this nematode? Well, for a few reasons. C. inopinata is morphologically divergent from C. elegans. It's massive compared to C. elegans, and also C. inopinata is gonochoristic, while C. elegans is a self-fertile hermaphrodite, so they have differences in reproductive mode. Additionally, C. inopinata is ecologically divergent from C. elegans. So C. elegans and other Cinerobditis nematodes have this sort of cosmopolitan distribution. You can find them in lots of different places, and they're associated with things like rotting plant detritus, flowers, soil, fruit, all these other things, but this sister species, C. inopinata, is associated with one species of fig in one part of the world and is associated with one invertebrate carrier, Ceratosylin fig pollinating wasp. So they're extraordinarily different in terms of their ecological niche as well. So suite of these things, the morphological divergence, the ecological divergence, the fact that they're sibling species yet are so different from each other, and they're Cinerobditis nematodes. They have tremendous power behind them as a model system and a tremendous wealth of background literature, make them an ideal candidate for doing the work that I do, particularly host microbe interactions. So as I mentioned earlier, these nematodes are bacterivores. They eat bacteria. And traditionally, they've been reared on E. coli in the lab. However, this is not what they eat in nature. But more recently, people have found wild microbes associated with C. elegans, such as Buck Samuel's group, Michael Shapir's group, et cetera. And so because of that, we sought to identify wild microbes that are associated with the sister species of C. elegans, C. inopinata, in order to investigate the evolution of host microbe interactions. And to do this, in 2019, Dr. Gavin Woodruff, the PI of the figworm lab that I'm in, he went to Taiwan and isolated about 45 microbial strains from the natural environment of C. inopinata. And in 2023, I went with Gavin to Taiwan and isolated about 60 to 70 more microbial isolates, so we have about 100, 110 of them. But in lieu of this work, what I've done is systematically rear C. naponata and C. elegans on some of these isolates in order to characterize their life history responses depending on what bacteria they eat and see where they diverge. And this sort of goes back to that first question in the slide full of questions a few slides ago. How do microbe-dependent life history responses evolve in divergent hosts? And I've done this, again, by doing life history assays with both species of nematodes, rearing them on different bacteria. And the first one I did was fecundity. So what is the fecundity of C. naponata and C. elegans when reared on fig bacterial isolates and E. coli? And the way I did this was by doing a standard cross where you put one virgin female and 4 reproductively mature males onto a plate with a given kind of bacteria, and then let them proliferate for a 7-day period. And after that 7-day period, measure how many worms there are on that plate at the end of a week. And I did this with C. naponata first with a lot of different bacterial strains, which is what this figure shows. So I want to direct your attention to the red dots right there. That is E. coli, and like I mentioned, this is sort of the ecologically artificial source, and I used it as sort of a control to measure the wild isolates against. And each one of these data points represents how many nematodes were on a plate at the end of a week for a given bacterial isolate, and this is for C. naponata, like I said. But I want to direct your attention to this isolate right here because it increased the mean fecundity of C. naponata by 103%, which is honestly extraordinary. And whenever we sequenced the genome of this bacteria, we figured out that it was a novel species, and so that's also really cool. But going forward in assays that I'm going to talk about in a second and ones that I'm not going to talk about during this talk, we decided to only use E. coli as a control and then this Klebsiella isolate because the response was so drastic. Our mindset behind it was that it'd be easier to tease apart the divergence of the reaction, the life history responses because of this. And also, I did this experiment with C. elegans, just with E. coli and Klebsiella though, and the results were recapitulated. So C. elegans reared on Klebsiella, our isolate, had increased fecundity in statistically significant manners. And I did this with 2 different genotypes. So the wild type, PT1074, and FOG2 as well. Because C. elegans is a self-fertile hermaphrodite, there's this gene FOG2 that, if there's perturbations in it, essentially makes the worm functionally gonochoristic. So I did this to control for the reproductive mode differences between C. naponata and C. elegans. But next, to tease apart this increase in fecundity in this first experiment I just went over, I wanted to measure the total individual reproductive output of these nematodes reared on E. coli and Klebsiella. So whenever I did that, I found something interesting, that for C. naponata, it doesn't matter what bacteria you are raised on, whether it's the Klebsiella isolate or E. coli, an individual nematode isn't outputting more progeny. There's no more embryos coming out. It's not statistically significant. But for C. elegans, if it's raised on Klebsiella, individual nematodes are putting out more embryos. So that's fascinating. And it poses this sort of question for C. naponata, though. Why is there an increase in fecundity in the first experiment, but no change in individual reproductive output? And so one hypothesis we had in this regard is that these bacterial isolates are influencing the developmental rate of these nematodes in different ways. So I measured that. And it's interesting because this scenario is sort of the flip side of the individual reproductive output. Because I'm interested in reproductive maturity, that's why I plotted this, but for C. elegans, it doesn't matter which bacteria it was raised on, it did not reach reproductive maturity differently depending on the bacterial isolate. But for C. naponata, there was a difference. If C. naponata was reared on Klebsiella isolate or Klebsiella isolate, they became reproductively mature about 1/3 of a day faster, which is 8 hours, which may not seem like a lot, but this is worm time, and they are extraordinarily fast, and you have to keep that in mind. So 8 hours can mean a world of difference in terms of reproduction for these nematodes. So sort of drawing it all back in to conclude, these are the questions we're interested in. I talked about my results from this first question, and our conclusions are as follows. So Klebsiella, our isolate, increases fecundity for both C. naponata and C. elegans. It increases individual reproductive output for C. elegans, but not C. naponata. And it speeds up the developmental rate of C. naponata, but not C. elegans. And perhaps most important, the evolution of a novel ecological niche, the fig that C. naponata lives in, has led to this divergence in microbe-dependent responses in terms of life history, and you can see this in different ways. So in the first experiment, both nematodes reared on Klebsiella had increased fecundity, but both nematodes are doing it in different sorts of ways. For C. elegans, it's just putting out so many more embryos, so that's why we saw that response. But for C. naponata, it's because the parents from the cross give rise to the F1s, and the F1s become reproductively mature way faster than if you're reared on E. coli, and then the progeny of the F1s, the F2s, then become reproductively mature way faster. And so that's why you see this more exponential increase as opposed to a slow increase with E. coli. So something's going on here, and I want to figure out what it is. And so I've isolated RNA from C. naponata and C. elegans reared on our Klebsiella isolate and E. coli, and I just sent those samples off like 2 or 3 weeks ago. So hopefully going to get a list of candidate genes, see what's going on there, and then do some knockout experiments with CRISPR to confirm the roles of these genes within these host microbe responses. And yeah, that's about all, but I want to give some acknowledgments to some people, some important people. Gavin Woodruff, Dr. Gavin Woodruff, the PI of my lab, and then Kim Mosher, our lab manager, lab tech. Without these people, I could not do what I do, and I feel so fortunate to be able to be in this lab. And also the School of Biological Sciences at the University of Oklahoma, as well as the NSF. Big thanks to the NSF for the funding. And then also John Wong from Academia Sinica for hosting us when we do our field work in Taiwan. And then also Sam Miller and Paul Lawson for helping us characterize the handful of new species that we have from our field work. And then Patrick Phillips for enabling the initial stages of this work in 2019 by allowing Gavin to go to Taiwan to collect these microbes. I'd love to answer questions if you have them. Thank you. Yes.
Yes, thank you. Great presentation. I was just wondering if your E. coli strains and the Ebola strains, if they grow in the same media, or
Yes.
if they have like different.
Yes, we're growing them in LB broth and then shaking them at 25. We're shaking them at 37, and we're ODing them to the same quantity, if that makes sense. So giving them the same optical density. So the idea is even if they have different cell sizes and such, we're putting the same amount of organic matter onto plates to control for the amount of food.
Okay, thank you.
No, thank you. Yes.
How do you make the C. elegans? How often, or is there?
How do you mate them?
Yeah, if you take take your mutants, do you mate them or keep them as hermaphrodites?
Well, so the wild type, they are hermaphrodites, and all you have to do is put one on a plate and they reproduce. But for the FOG2s,
Yeah.
there are males and females of the hermaphrodites, and so those just mate together and remain.
But how so they are like co-cultured?
Yes.
There's no specific
Yes.
mating protocol.
Yeah. You just put FOG2 on a plate, the one that has that mutation, and then it maintains the mutation, because it's knocked out. Yeah. Yes.
When you look at the population structure of all the ornaments found in Taiwan, and how does that compare to the Japanese isolates? And whether they respond differently to your new bacteria?
I have not, but that's something I'm really interested in and I've talked about with Gavin. It's something that we're interested in looking into, but I guess I don't have because there are wild isolates like the Ishigaki strain and stuff like that, and the Okinawa stuff, so yeah, but I don't know.
So, hi everyone. Thanks for being here. I'm Richard Maitern from University of Tartu, Estonia, and today I'm going to talk to you about some of ongoing research that I'm doing on life history traits and toxic and sensitivity on fish. So in our lab, we are really interested in why some species are more sensitive to toxic substances or environmental pollutants than others. So this is what I'm going to talk to you about. So as you know, different species react differently to different environmental pollutants, but the question is why there is such a difference? Or maybe some species groups are better adapted to these pollutants that come into the environment, just because of their evolutionary background, or maybe there is this, they have been evolved in some situations, in some environments where they just need to be more tolerant to toxic substances. And in ecotoxicology, there is this thing called species sensitivity distribution, which you see in this picture here, that there is a certain chemical concentration, all species go extinct. But there is a gradient for each chemical or a toxicant in the environment that affects different species differently. So in these species sensitivity distributions, they try to find out this kind of concentration, which is like acceptable in a way. It's usually 5% or something like this is considered like, okay, 5% of species go extinct, and this is, or 5% for 5% of species, it's not that bad. So, but in ecotoxicology, we also want to know why this happens. And there is like two general terms here. The one is, first, when a chemical comes into the body, you either don't let it inside the body, or you get it out really quickly. So, and this is the toxicokinetics part of the defense system of a fish or whatever. And there is also this toxicodynamics part where just enzymes evolve so that they are not so susceptible to these chemicals. So this, to bring you an example of this toxicokinetics, which is, I think, easier to bring an example of, there is this one nice study done in Galveston Bay, where there is this species Gulf killifish, and there is this huge gradient of pollutants that near the river there is maybe I can show you. Okay, anyway, the black dots, there is a high chemical concentration of these polycyclic hydrocarbons, PHAs, and there is this difference in populations where these populations are more adapted to this, and this has happened within 60 generations. So in this Gulf killifish, and we see that they have actually downregulated this AR or aryl hydrocarbon receptor pathway. And how they have done that is actually these genes have gone from another species that is some 2,000 kilometers away, and genes from their population are somehow they have been mating. So got this kind of a pollution protective gene into their genome. So what we wanted to do was we wanted to see if there is a phylogenetic structure to toxicant sensitivity in fish. So is there a phylogenetic, and we also wanted to see if there are certain life history traits that correlate with higher or lower toxicant sensitivity. So if there is we can see it inside somehow the environment affects this. So what we wanted, we first looked at what has been done before, of course, and there are usually these species sensitivity distribution studies, they usually, in minimum, they use 7 species. So which is not that much if you think of you want to characterize a chemical. But there are a few that have been done on more species, but they are really looking at a few selected chemicals. Like here I have been brought out this cadmium with 27 species, and then this chloropyrifos in a lot more species, just to see. But this is more like to see a specific chemical effect but also, and these species sensitivity distributions, they usually use more species, not just fish. But there is also one study that has looked specifically on few fish species, and what they brought out that salmonids are more sensitive to chemicals, and also that metals are important in this context. So we took actually the same database that the previous study used, but just at the later point of time, and we used more species. There is this Ecotox database in the US governmental or environmental agency Ecotox database that has this lethal concentration 50 data for a wide variety of species from very different backgrounds. And we didn't take all of the chemicals from there, but we went for a specific list of chemicals that have been said to be environmentally important. So these are like defined as in the water directive in the European Commission to be. So there are 29 chemicals, and they were into these classes, into these chemical groups. So what we extracted from there was around 4,000 rows of data, and the chemicals and the species there are not equally distributed by far. There are a few that are really studied, like discadium or some BH8Hs like this pentachlorophenol. And we also wanted to make it more, and we also tried different filterings on the data because it's not totally homogeneous. So we tried to have at least 3 different studies on one chemical or a species group. So, and not to compare apples with oranges, we had to somehow standardize all the chemicals that we are comparing. So what we did was we calculated per chemical across species what is its impact on the species group. So this is a way how to try to put the things together. And we also for the life history data we gather from FishSpace and other databases, different physiological traits that seemed relevant or were fairly easy to gather, to include, to try to see whether there is a migration difference or feeding type or any of these things affect. So in the end we had 32 orders and around 250 fish species. So, and this is the results where we took all the chemicals together, and we had the order is in here in the and this is a Bayesian model with species as a random effect, so no phylogeny is taken into account. And here we see some differences between orders, and that sturgeons or herrings are different. But once we take the phylogeny into account, the credibility intervals go a lot wider. So there is no clear-cut trends here that we can actually see. Still, the salmonids give also a broad range, and so this actually happened with all of these, whether we looked at families or orders. And when we've also looked at with specific compound groups, we also saw that there is a huge variation between within these orders or families, fish families, so that there is the credibility intervals are so high, so wide, that there is no clear differences. So, and actually the same thing happened with all of the variables that we looked at life history variables. Here is air breeding, which is something that we first was thought that the same thing when we had the species as a random effect, and when we had the whole tree or the phylogeny included in the model. So why? Why is that there is a huge difference in the without phylogeny and phylogeny models? And when we did also some simulations, but maybe this is best seen on this part of the tree that we have in the analysis. So there are two species that are closely related. The blue is now here, as in the previous air breeding, and the red is non-air breeding species. And there is a huge difference within closely related species. So, and when we have below here, below the Shanna family, you also see that there is this salmonids. And the salmonids are a group that really show a really clear trend of on the same side, that they are all fairly sensitive to pollution. But they are also really, really evolutionary close in that respect. And you see that the tree has this branch lengths. So this is, and this Channa, if you think back to the study that they had, it's also two different species. That one is really well adapted to the pollution, or is really good handling the pollution levels, and the other is not. So when a gene from the other sister or related species comes into there is this, it brings benefits to other species. So there is no clear, so maybe looking at phylogeny in these kind of analysis is not the best in that respect. That how do I say it? That they are so evolutionarily new, some of these chemicals, that there is no pressure, and even the previous levels of these chemicals were so low that there is no groups that are better or worse protected. Yes, so thank you. Any questions, you can go. Yeah.
I'm interested in if, could you name or think of any like ecological factors pre-industrialization that might have predisposed some species to toxicity tolerance over others?
Yeah, if you think there has been pollution around, or these chemicals have been around for a long time, and we also try to separate for old chemicals and new chemicals in our analysis, but we didn't find a clear trend there. But definitely in the ocean bottom there are some deposits that are good for most of the fish. Or if you take a benthic fish, for example, they might be exposed more to some kind of chemicals in some parts of the world. So there could have been something, or there could have been something because of, but right now, yeah, we can't see it. Yeah, okay, thank you.
So good afternoon, everyone. My name is Sophia. I'm a master's student at Concordia University here in Montreal, and today I'm going to be talking a bit about my thesis work on the effects of environmental variation on maternal life history responses in divergent brook trout populations. So environmental variation refers to changes in abiotic conditions like rainfall, seasonality, and temperature, but as well it refers to changes in the biotic conditions like trophic level interactions and food web dynamics. This variation drives life history evolution, and it influences changes in an organism's lifespan or reproductive strategy. For fishes, when they're adults, this can mean changes in their somatic growth rates, their size at maturity, their fecundity, and their egg size. And in particular, when they're in their juvenile stage, it can have consequences on their growth trajectories and overall lifetime reproductive success. So how does environmental variation influence maternal life history trade-offs? In resource-rich environments, we know that females are going to invest a lot less energy into their offspring to produce a lot of eggs at a very small size. They're going to mature earlier at a much larger body size and allocate a smaller amount of that body mass towards their gonads. So they're going to prioritize their own growth over their reproduction. And in resource-poor environments, we expect to see the opposite, where females are going to put a lot more energy into their offspring rather than their own growth. While many studies have examined these evolutionary trade-offs, the relative extent to which the environment and intraspecific variation influences these trade-offs is an ongoing area of salmonid research. And you might be wondering, well, why salmonids? Well, they're a really ideal group of fishes to examine the extent of intraspecific maternal life history responses towards environmental variation because they're locally adapted to their unique conditions. So a lot of their population level responses are going to be diverse and variable, and that's led them to persist and adapt despite all of these changing conditions. So intraspecific population variation occurs through local adaptation, parental conditions, and phenotypic plasticity. Many populations of the same species are living in different habitats, demonstrate extensive variation in their life history responses. So here, these are 4 populations of brook trout that occupy an environmental area of about 25 kilometers squared, but they have really different body sizes, coloration, and morphologies. So it's important for us to be able to understand the extent of these responses to be able to forecast how these species are going to respond themselves towards change. This leads us to my main research goal, which is how do maternal life history responses change towards environmental variation across divergent brook trout populations? And when I talk about environmental variation here, I'm referring to food availability, which is something that we can alter and manipulate in the lab at Concordia. So we predicted in resource-rich environments that females from all populations are going to maximize their fitness by investing less energy into their offspring to prioritize their own growth. So they're going to have a higher fecundity but smaller egg sizes. In resource-poor environments, we would expect the inverse to happen, but we would still see that it would have the largest impact on fast-growing populations than slow-growing ones. Our study uses 4 genetically distinct brook trout populations from Cape Race, Newfoundland, which is a really cool area. It's been well studied since 1990s, and the populations here are genetically distinct, and they're all geographically isolated from each other. So you can see these are some of the rivers that these fish live in, and all of them end in either a waterfall, off a cliff, or into the sea. And these populations are all landlocked, so they live basically in an area that's about 2 kilometers long, and they exhibit life history variation akin to what we would see across vertebrate species groups, not just for one single species. Past research has assessed that each population has distinctive genetic growth rates, so that's how we've classified our 4 populations as fast growers, intermediate growers, or slow growers. And we've done this because we wanted to be able to see a spread of genetic and phenotypic diversity to really explore intraspecific variation. So our experiment technically begins with wild gamete collection in Cape Race in October of 2021. We flew those gametes to the aquatics lab here in Montreal, where we generated a large number of full sibling crosses, and we reared them under common environmental conditions until our day 0, which is marked at the post-yolk absorption stage, or the phase where fish begin to swim up, they feed by themselves. However, they're still not big enough to actually run experiments on, so we had to wait until July of 2022 when fish enter the early juvenile stage. So from the early juvenile to late juvenile stage, this is a pretty important season for the fish because it's their first time where they'll be able to choose, technically, whether they're going to reproduce or not that year. So all these acronyms here represent the population, and each circle is a tank. We had 4 populations, and they were reared individually by themselves, but we had 10 replicates for each for each treatment. So our treatments were a high food or low food, and high food represents fed to satiation, while low food was 1/3 of this amount. At the late juvenile stage, fish enter their second growing season until they reach maturity. And it's at this point where, due to space limitations, we had to start pooling populations. So what we did was pit tag them, or insert a little chip that allows us to identify the fish at the individual level, so we know who's who. And then we had 2 replicates for each experimental treatment group. So to look at our experimental treatments, from the early to late juvenile stage, fish are fed either a high food or a low food diet. And then from the late juvenile stage to maturity, they were either kept on the same treatment or switched to the opposite, which gives us 4 experimental treatment groups, a high-high and a low-low. That's our resource-rich environment and our resource-poor. And then a high-low and a low-high, which are our reversal of environmental conditions. After 558 days in November of 2023, fish reached maturity. This is when we can actually measure those maternal responses, and we took metrics for size at maturity and growth rate, as well as fecundity and egg size for around 400 fish. And to sort of highlight our main results, we're looking at the resource-rich, so our high-high treatment versus our resource-poor, our low-low treatment. And on the y-axis, we've got the mass, and on the x-axis is in days. So when we look at whether late juveniles were able to, what their growth rates look like during the stage, we see that fast growers grew fastest than all other populations, and slow growers always grew fastest in the high-high treatment than any other. We see a similar trend when we look at these box plots, when we look at body size at maturity, where fast growers again are our largest across all treatments, and slow growers were always largest in the high-high than in the low-low. Similarly, when we look at fecundity, we see that fast growers had disproportionately high amounts, high fecundity, and slow growers always produced more eggs in the high-high treatment than in the low-low. However, when we look at egg size, we see that only population influenced this response, and that was only between fast growers and slow growers, not any other populations. And we see that egg size remains similar across all our treatment groups. When we look at the reversal of environmental conditions, our high-low and our low-high, we see something that we didn't expect, which was that all the maternal life history traits were similar regardless of the treatment that they were in. So high-lows and low-highs, didn't matter what the response was. These fish were always most similar to each other in these groups, which was quite interesting. So to conclude, in resource-rich environments, we see that our first prediction that all females would grow fastest, but that would be disproportionately true for fast growers and slow growers. We found evidence to support this. We also found evidence that females would be largest at maturity when they're in resource-rich environments, and that these females would have the highest fecundity. However, we didn't see females produce smaller eggs in this environment. And one of the reasons that that might have been the case is that even though these populations demonstrated extensive plasticity across their fecundity and their growth rates, maybe not all these traits are not as plastic as we anticipated them to be. And for egg size, it might be that it's genetically constrained and that the influence of environmental variation is masked by this genetic influence. And when we take a step back to look at the bigger picture and the consequences of this research, it's that healthy salmonid populations here in Canada are indicative of healthy ecosystems because these fishes are really important for our commercial fishing industry, our recreational fishing industry, and as well as a lot of Indigenous groups. So we want to be able to look at them and explore the roles of genetic and phenotypic plasticity of all these populations towards ongoing environmental variation so that we can help to conserve them for future generations of not only people, but also fish. So I just want to say a huge thank you to all the funding organizers, as well as ACF at Concordia that reared all the fish, and the members of my lab who made all this research possible. Thank you. Yes.
Thank you, Sophia. A great presentation. I was just wondering, does your high and low nutrient treatments are based on the nutrients that the natural population have?
No, not at all. So the reason these fish are really interesting is that because they occupy streams that are really food limited, and in the wild they're actually only going to be about this big, but when we grow them in the lab they almost triple in body size. So I guess I should have mentioned that, which is why we wanted to see high as a true, like, let's feed these fish as much as we can so that we can get them as big as possible to see just how extensive their body rates can go in a captive environment. Yes?
So, just taking off on that, just what you said, if they're in very food-limited environments, then it kind of makes sense that there would be stabilizing selection on any size, right? That it's really important to have big enough progeny to be able to feed or hold off and survive long enough to get to food. And then, presumably, like my experience with chars and whitefishes is they would just add eggs to the train. They have
Good to know.
But,
Yeah.
I mean, I think it's very nice work, very nice. I really like when people test ecological things by experimentation.
Thank you. Yes.
Somewhat related to that, so you showed the contrast among the different treatments and how that affects egg size. How much variation is there among individuals with egg size and fecundity? Can you parameterize that relationship, the fecundity and egg size relationship?
That's a great question. Yes, and actually, when I was talking to my supervisor about all this research, that's the next step for this, is to look at each individual female, how different are their egg sizes within themselves, and what that variability might look like. So yes, ongoing area of what we want to look at.
More granular look at it.
Yeah.